The overall goal of this procedure is to knock down endogenous gene expression by injecting Drosophila embryos with double-stranded RNA to identify genes required for organ development. This is accomplished by first collecting and lining up embryos in a straight line. Next, needles are prepared and used to inject double-stranded RNA into embryos. Finally, embryos are placed into a moist chamber to develop. Ultimately, results can be obtained that show defects in Drosophila tracheal branch fusion through antibody staining of late-stage embryos or by observing larvae under a bright field microscope.
The main advantage over this technique over existing methods like forward genetic screen to identify genes required for organ development is that this is a relatively simple and quick reverse genetic approach to analyze gene functions before a mutant is available for that gene.
This method can help answer key questions in the developmental biology field, such as the identification of critical genes required for the development of the Drosophila tracheal system. This is an excellent model to study the morphogenesis of mammalian organs, such as the vertebrate airway, circulatory system, kidney ducts, and excretory epithelia.
Generally, individuals new to this method will struggle because it takes time to learn the methods such as lining up embryos, breaking needles, and injecting double-stranded RNA into the embryos. Visual demonstration of this method is critical as the steps involving the amount of double-stranded RNA injected varies between embryos, and written demonstration of what syncytial blastoderm embryos look like can be difficult. These steps are difficult to learn because injecting suitable amounts of double-stranded RNA into syncytial blastoderm embryos is such a critical step to effectively knocking down the expression of endogenous genes.